This is Robert Sapolsky, and you're listening to Father-Offspring Interviews.
Hey everyone, welcome back to Father-Offspring Interviews, episode 65. We've officially passed 30,000 subscribers on YouTube, so we wanted to take a moment to thank you all for your interest in science and why it matters. We love that there are people from all over the world on this channel. That's something that we very much hoped for when launching this series. And we really do read all of your comments and questions and enjoy seeing names that have become familiar by now, so thank you.
Yes, thank you.
We are going to start off with a question from Sabrina from Montreal, which is, how come humans and chimpanzees share almost all of their genomes, but humans are so different from chimps? Where does the 1% difference in genomes lie?
Oh, I love this topic because it's got an answer that is so interesting and elegant and explains so much about the universe. Humans and chimps, as you heard, we share 99% of our DNA. Bonobos, they share 99% with us as well and with each other. And just as a proviso, 99%, any percent is a bit of a squishy number because it's surprisingly undecided upon exactly how to count how many genes there are, but something in that range. We and chimps are almost genetically identical. So 99%. And to try to put that in perspective, you then go through other living things and we share a smaller percentage of DNA with them. For example, about 94% of our DNA we share with baboons. We share about 90% with a cat, about 80% with a cow. We share about 60% of our genes with bananas. So that raises a puzzling question. And this is one that we've addressed before. Okay, so you share 60% of your genes with bananas, but if you pay attention to sort of sociobiology type topics, you will know that you and your full sibling share 50% of your genes. What's up with that? Are you more related to bananas than your full sibling? What we have is an unfortunate convergence, the same way of describing two very different things. We share 99% of our DNA with chimps, 60% DNA genes. What kind of traits are they coding for? In contrast, within a species, you and your sibling within that gene, what variants of that type of gene are you sharing? For example, you look at bananas again, and we have genes for nose hairs, and they don't. Bananas don't. And bananas have genes for looking like banana shaped fruit, and we don't. And types. Nonetheless, we and all other humans share having the type of gene for nose hairs, and it's probably not just one gene, but it can come in different variants, and you can have luxurious curly, and you can have spiky and dangerous, and you and your full sibling have a 50% chance of sharing the variant of this type of gene. So, given that 60% is not that surprising, shared with banana trees or bushes or whatever they grow on, because plants have mitochondria and we have mitochondria and they need powerhouses of cells and, they got DNA and it's so there's lots of similarities. So, this gives us a feeling 99% with chimps, down to 60% for bananas. So, thus, back to this question, what do we make of that approximate 1% difference between us and chimps? And this is where we get this wonderful, subtle concept coming in as follows. You take a neuron and it's from a shrimp, it's from a baboon, it's from your neighbor, whatever. And it kind of works the same way. It's got the same ion channels, it does action potentials. There's remarkable similarity A neuron is a neuron is a neuron from all these different species. You put them under a microscope, unless you're super specialized, you can't tell the difference. They look the same, they smell the same, they taste the same. Neurons are a basic unit. We've got neurons that are not a whole lot fancier than neurons you would find in a starfish. What's the difference? Not that we have fancier neurons, we have more of them. Okay, to give you a sense, tardigrades, those are those water bear microscopic thingies that are like immortal and they're incredibly cute. And you would want a tardigrade stuffy and a tardigrade that, I don't know, mostly does this in water. What a tardigrade has to work with is 200 neurons. Throw 200 neurons together and you can get a tardigrade. Throw 1,000 neurons together, and you're working with a level of complexity that a starfish could come up with. 1,000,000 neurons, we're talking cockroaches who can learn stuff. 1 billion neurons, and suddenly we're in the range of like a horse. 10 billion neurons, a baboon. 30 billion, approximately, a chimp, a bonobo, a gorilla, and then us clocking in at around 80 billion neurons. Just before we get all high and mighty about having so many of those neurons, there's some species of whales that have more neurons than us. They get up to the 100 billion range. Elephants have 250 billion neurons or so. And how they count in all these cases, who knows? But okay, we're not the best in terms of the most neurons, but we're way up there. And thus we have this puzzle. You put 30 billion neurons together, and it forms something that could do what chimps can do. And it could make tools, and it has rudimentary culture. And as we heard the other week, it might have the starts of syntax and chimps, and they can do organized warfare. And then you put 80 billion neurons together, and out pops a species that can invent 6,000 different religions and 7,000 different languages and economic systems and superstition. And what we have here is this really important concept in this field of emergent complexity. You put enough simple building blocks together, and out of that can emerge totally amazing things. And two sound bites describe that. One is more is different. As you have more simple elements thrown together, stuff emerges from that that is not formally predictable from the level of the individual building blocks. It's only describable at this upper level. And what you see as the other soundbite is sometimes with enough quantity, you invent quality. So, what would be an example of an emergent property? A molecule of water, H2O, cannot feel wet. The property of wetness is an emergent attribute of lots and lots and lots of molecules of water. And as soon as you got enough water molecules together, that wetness has emerged. The water molecules are still just as stupidly simple as they don't undergo any special changes in order for wetness to emerge. Just get enough of these together, and this wetness thingy emerges out of more is different. With enough quantity, you get high quality wetness. And in the same way, what we're seeing is something along the lines of neurons. And neurons, basic, simple building blocks. They're not simple, but compared to the whole thing, they're basic constituents, and they're roughly similar in a tardigrade and in a giraffe and in us. And all we're seeing is throw 200 of them together, and out of that emerges, without a blueprint, and without a blueprint maker, the level of complexity that can keep a tardigrade in business. And the numbers scale up from there, and throw 30 billion neurons together, and you got something that has the properties, the emergent properties of a chimp brain. And throw 80 billion, and now you got a human brain. So, what's the 1% difference? People have known this, and it's 1 to 2%, depending on that squishy definition again. People have known this for a while, and as soon as they identified the 1 or 2% that was uniquely human, everyone went crazy. What are the differences? Because that's going to explain us and our magnificent cultures and chimps just coming up with hitting sticks on termites and things like that. And it was mostly disappointing. About half the genes that distinguished us from chimps had to do with olfaction. Chimps are not very olfactory creatures compared to, say, dogs or rodents, but they're a lot better than us. And about half the genetic differences are genes coding for olfactory receptors in chimps that have been deactivated in us, called pseudogenes. Whoa, that's half the difference? A big chunk of the remaining ones had to do with body hair. Why it is that it's only a rare human who has chimp-like fur on their shoulders. That's some immune stuff, some stuff making humans and chimps not particularly reproductively compatible. Where are the genes that differentiate the chimp brain from the human brain? Are there any genes that are, for example, only found in humans and only active in the human brain? And there's a handful now, and they explain everything. Now, the gene that's arguably gotten the most attention is called ARHGAP11B. And usually these things get some flashy name. So I don't know if this is called an R gap or an R gap, but in any case, this is a gene that's only found in humans. What does this gene do? Something really interesting. How does your cortex wire up during fetal development? It's this two-stage process that you first have these glial cells. Remember, glial cells are the unsung heroes of the nervous system. They're nowhere near as interesting as neurons. You've got glial cells, and what happens first is what is at the base of what's going to become the cortex. Glial cells migrate to different spots here, and then they start growing an axle, a long stretch of cell that goes up to what will be the surface of the cortex, radiating upward. And these are called radial glia. They are the scaffolding. It's a very different way that scaffolding is used than with human construction projects. You got a building and you want to renovate it, so you put up scaffolding around it. In the case of the developing cortex, the scaffolding, the radial glia, come first. So there's a whole array of them, and neurons start migrating. And the rule is if you bump into a radial glial cell, you begin to grow up it all the way to the top to the surface. And the next neuron that comes along and bumps into it grows up and stops here, and you form a column of cortical neurons. And this is going on with all the radial glia. And this is essentially how you form a cortex, this two-step process of glia optimizing the shortest possible path from the bottom of what will be the cortex to the top. What that tells you is there's a bunch of radioglia that get uh, actively killed during development because they've got an inefficient projection winding all over the place. You optimize the shortest possible pathways to the surface, and then migrating neurons wander randomly, and their only rule is that you bump into a radioglia and you start migrating up it until you bump your head into the rear end of the neuron up above you, and you begin to form these functional columns of the cortex. So that's how the cortex wires up. What does R-gap do? What it does is it increases the number of radial glia made in the developing cortex. It boost up mitochondrial activity in glial cells. So they've got so much more energy, since we all know mitochondria are the powerhouses of the cell. So mitochondria are pumping out more energy, and this makes the precursors to these glial cells say, wow, we're so energized. Let's make more radial glia. And so you've got a lot more scaffold, and you wind up having more neurons forming there merely by making more of the scaffolding, you will wind up with more neurons. And there's now a small family of genes that have similar properties to these, and they're all active during brain development. They are all found only in us and not in chimps. And what they result in is more neurons. Whoa. What are some of the implications of this? Some researchers have followed up on this, and there's some plenty interesting ones. They've taken the gene for RGAP11 beta, they've taken the gene for that, and they've stuck it in mice, and they've stuck it in marmoset monkeys, and they've stuck it in rhesus monkeys. And these are animals who get more neurons in their brains. Sticking in various members of this family of human specific genes, you wind up having animals with a bigger cortex, more neurons there. And of course, what you're asking right now is, do they get smarter than your average mouse or marmoset? And the answer is yes. So we have a really surprising, simple answer. How do you turn a chimp into a human? What's the 1% difference? Just have a handful of genes, which during brain development says, let's make more radioglia so we'll have more neurons, and let's have neuron precursors divide a few more times. And in order to turn a chimp into a human, just have a few more rounds of cell division and a bunch more radioglia to provide scaffolding. And if you did that to a chimp, and you boosted it up to it having 80 billion neurons, that chimp would invent theology and language and art, and it would invent totally different kinds, no doubt oriented towards bananas. But what you would get there is more is different. With enough quantity, you invent quality. So it takes a remarkably small number of gene differences between us and chimps, just double, triple the number of neurons, and that's a very different primate that's going to emerge from that.
Okay, next, Claudia from Romania asks, can you become addicted to stress hormones?
Yes. Okay, we're almost a century into the concept of stress being bad for your health. Stress damages this, damages that, accelerates brain aging, does everything bad. And thus, it's easy to reach the conclusion that stress is uniformly bad for you. And all you need to do is reflect a little bit and realize no, there's some types of stress that are not aversive. And in fact, we love them. We love them. We pay good money to be stressed in just the right way. What is it about good stress that has the potential to become addictive? Okay, you have this concept we've had before, the inverted U. Here is the amount of stress which is perfect. It makes you alert, it makes you happy, it's stimulating. It's stimulating. And what you see is showing this inverted U, too little of this type of stress, and you're bored. Too much, and you're adversely stressed. Getting an optimal up there. Certain characteristics of what type of stress wind up being optimal. What is it that makes stress appealing? It's not too severe. It doesn't go on for too long. It's with an overall setting of feeling safe. And that's what play is. What are two dogs doing when they play with each other? One of them crouches down, the higher ranking one, and is communicating, I am suspending our dyadic rank differences here. I'm suspending it so we can just run around like crazy and pretend to knock each other over. And I can put my teeth all over your throat, and it's now a benevolent setting in which they can surprise each other. What is it that we call good stress? It's when it's stimulating and it's surprising. And it's surprising in a way that doesn't carry menace. Okay, so you've got an optimal level there. So, how do you get around to this optimal level? And one of the things you see, which is extraordinary, is you take rats and they can do what's called a self-administration paradigm. The rat has to work. The rat presses a lever, and by working, it presses the lever some number of times, and it gets an infusion of a drug. What drug are we talking? It gets an infusion of a drug version of stress hormones, glucocorticoid hormones like cortisol in humans. Rats will work. They will work to get infused with glucocorticoid stress hormones. How high? Just up to the optimal level. Just for them feeling they're most alert, they're most great. And what are those levels of glucocorticoids doing in the brain? They make your synapses get excitable more readily. It increases the amount of glucose getting into your brain, the amount of oxygen. It's absolutely great. It's optimal. And very much as part of that story, it also causes the release of dopamine and parts of your brain related to pleasure and anticipation and goal directed behavior. So, what we're seeing here is a rat will work to get infused with just the right amount of stress hormones to optimize its amount of dopamine release, to feel maximally stimulated. And that's when it's working well. Okay, so where does one begin to get into trouble here? The first obvious thing is people all differ as to what their optimal set point is. Their optimal set point of glucocorticoids in their bloodstream, or their optimal set point of feeling terrorized by a scary movie, or people have different optimal set points, and one person's optimum may be playing shoots and ladders, and somebody else it would be chess boxing with some big beefy Russian champion, and people have different set points. So once you've figured that out, okay, what every person wants to do is optimize their level of stimulation and unpredictability and lack of control, the things you give away in play, that you give away when you're excitedly going to be surprised by an unexpected ending that's not menacing, all within a benevolent setting, doesn't go on for too long. Again, those same parameters, that everyone has their own optimums to what they want to achieve, where the stress, in that case, the challenge, feels great. So, where do you get into trouble? At least two different ways. The first one is if you inadvertently overshoot and you've gotten yourself into a circumstance that is not optimally stimulating, and instead you blew it and it's way over in the stress, and you decide, oh, I don't know, I keep reading about how learning a new language is good for your brain, so I'm going to learn Uzbek, and you sign up for Intro Uzbek, and everybody else in there is fluent already, and they're all arguing about grammatical rules in it and scimitars, and I don't know if they have scimitars in Uzbekistan, but and you're getting a grade and everyone is way ahead, and you are way past the point of, oh, it'd be cool to learn some new words in Uzbek, and instead you're way down at the strip. You can overshoot it. You can undershoot it and then you're bored, but the overshooting is much more damaging. Okay, so that's one way in which things can go badly. The second is, as you go through this, as you stimulate your world to get to these optimal set points over and over, the set point can change. The set point can become less sensitive. What's that about? That's the thing that was so amazing and exciting the very first time you did it. You do it today, and you still like it, but it's never like the first time. It's never like the first time because set points habituate, desensitize, they shift in a direction, just as with drug addiction, where you need a stronger and stronger oomph to get up to that optimal point. And that's exactly the realm in which stress hormones could be addictive, where you need to have a more extreme challenge in order to get to that optimal point, because yesterday's version still has you at the boring end of the inverted U. And I don't know, Claudia, Romanian has this sort of idiom, but in English, it's the notion of being an adrenaline junkie. You get so habituated to this need for this burst of scalding hormone that's telling you emergency and challenge, and maybe you could do it, and it's going to feel so great when you do it, but you're not completely certain, and that's great. And tomorrow, it needs to be a little bit more extreme. So, in that regard, it is addictive like all standard classical drugs of addiction. And rather than being adrenaline, the most famous stress hormone, it is much more a case of being glucocorticoid, cortisol junkies.
Finally, Edith from Philadelphia says, this question is following up on the discussion of GLP, GLP-1, and semaglutide-based drugs in episode 56. So, if it is true that the body can't differentiate between different kinds of physical and metaphorical hunger and satiation, is it also possible to lose weight by engaging in activities that might increase satiation of some kind that's not food related?
Yes, harking back to episode 56, uh, what is satiation about? You consume a bunch of food, and because of the nutrients being absorbed into your bloodstream, the distension of your stomach and your intestines, you release a bunch of hormones that get back into the brain and give a signal of satiety. And one of the hormones is leptin, that used to be very flashy and is now tragically ignored. But the one that's getting most of the excitement these days is GLP-1, glucagon-like peptide 1. And it's released from the intestines in response to a meal, and it gets into the brain where it makes you eat less. Okay, let's deconstruct the makes you eat less. One of the important things is it goes exactly where you would expect it to go, which is it binds to receptors in the hypothalamus, neurons there that control feeding, and it inhibits them. But what people were very surprised to see was it was binding in all sorts of other places in the brain, in places where there are receptors for things like GLP-1, unlikely places like your hippocampus, which has something to do with memory, your olfactory bulbs, having to do with olfaction, back to chimps, your ventral tegmental area, which is involved in dopamine synthesis and release. And this began to explain how much satiation is so much more complicated than your intestines sending a signal, we're full, we're distended, we've got all the nutrients we need. And this has come to be appreciated with the synthetic versions of GLP-1, the semaglutides, like most famously Ozempic, that are revolutionizing weight loss, and they're doing amazing things. They are working in the hypothalamus to suppress feeding, but they're also working in these other areas like the olfactory bulb to make food smell less appealing, in the hippocampus, so you're not having as strong of a memory of food craving, in your dopamine area, so you're not having as strong of anticipation. And it's thus this hormone that after the fact, after you're eating, Ozempic makes you feel full faster than would otherwise be the case. But beforehand, it's decreasing the craving, it's decreasing the hunger for it. Okay, so that was the basic Ozempic story. And out of that has been emerging a remarkable finding in recent years, which is alluded to in the question, which it turns out people who, for example, have a gambling addiction, Ozempic lessens their craving for gambling. Ozempic helps in that realm. Ozempic helps with a bunch of addictive type behaviors. Whoa, that's totally weird. It's this hormone that's in your hypothalamus and it's telling it about blood glucose and stuff. What's it doing with gambling? Out in your hippocampus, out in your dopamine anticipatory areas. And what we begin to see, what I emphasized back in that episode, is we start with a metaphor. Ooh, I have a hunger for food. Okay. That's kind of literal, but you can have a hunger for success, for money, for love, for status. No, 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 no. Those are just metaphors. What it tells you is all of those yearnings and cravings that we have overlap in some ways with the neurobiology of plain old, very simple food hunger. And things like a hunger for a better world or a hunger for a new pair of socks or something can tap into some of the same pathways. And thus, Ozempic seems to be having this ability to damp down all sorts of our hungers that we are driven by because they are not easily sated in us. Okay, so this brings us to this question now, because what that begins to suggest is, ooh, hunger, hunger for money, hunger for food. It's not just a metaphor in the hunger for money, love, status sense. It's using some of the same neurobiology. And to bring in a concept here that is very, very metaphorical, all these different types of hungers are using some sort of common currency. And thus the question, if you are having way big of a deficit in fulfilling your craving for X in your brain, can you decrease some of that craving by satisfying your needs for Y? Will they crosstalk? Will they crosstalk such that having particularly sating, satisfying stimulation in a non food realm makes it easier to not eat? And this notion of crosstalk between these two systems is something that was pioneered by this wonderful scientist who unfortunately died early in the pandemic, Mary Dahlman, one of the pioneers, great person, University of California, San Francisco Medical Center, who showed why it is that when we are stressed, something very deleterious happens to our health. Now, it's great if people get cures for COVID and cancer and all these things. You want to go solve more ill health on this planet. Go figure out why it is that when you feel like nobody loves you, you eat more M&Ms. Do that, and you're better than Jonas Salk. And that's what she began to figure out. She showed two things. When you are under stress, glucocorticoids get into your brain and work in the hypothalamus to cause you to have a food craving. And not just any food, but the worst food for you, high fat, high carb foods. That's where you get the munchies from when you are stressed. That's where you get comfort eating. And then completing the loop, when you eat those foods, they decrease the release of glucocorticoids. So we have a crosstalk between whatever will cause you to secrete glucocorticoids. You have hungered for winning your chess boxing match and it hasn't happened, and you are unsated and you are secreting glucocorticoids that are having adverse effects on you and making you feel crummy. And they're causing a craving for high fat, high carb foods. And when you eat it, you shut down the glucocorticoid release that was caused by your chess boxing trauma. Thus, we have the idea proposed here, which is in a circumstance, if you have someone who is having trouble controlling their eating, if you satisfy some of their other hungers in a dramatic way, the crosstalk, the decrease of glucocorticoids because they're satisfied in this other realm, less glucocorticoids, less craving for the food that's bad for you. And okay, this sounds very abstract. You get into like a flow state, one of those, you're sitting and you're working on something that's engrossing and it's pleasing and it's stimulating, and you're focused and your sense of time completely disconnects. And before you know it, you've been there focused and satisfied for 7, 8 hours, and you didn't even notice you had just missed 2 meals. That's where satiation in a very non foodie part of the brain, you're sitting there solving Fermat's last theorem, and you do it in 7 hours, even though it's been done already. And that is very satisfying. And the process is very satisfying. And you are engrossed. It's not just distraction. The intensity of satiation there is overlapping with the traditional food satiation pathways, and you are not stimulating as much of those. I'd love to have some chocolate covered potato chips right now, and thus the food craving goes down. In principle, it should work that way, and we know it does in circumstances like that.
And that's it for episode 65. Keep submitting questions at the form found in the Instagram story highlight and bio or the YouTube video description. I'm Offspring Sapolsky, and thanks for your continued support of science and the beard.